Good afternoon, everyone. I'm Joe Barone. I'm the Dean of the Pharmacy School, the Ernest Mario School of Pharmacy, which, as you know, is right next door. And it's my pleasure to introduce today's symposium. I just want to give you a little bit of background on how we got here. I encourage you to, I won't bore you with all of the details, but just take one of the flyers. Yes, I took it off the board, sorry. And you get a little bit of idea of what we're trying to do today, but just a little bit about how we got here. Our two speakers, J Augeri and Brian Strom, are both on our faculty. Before joining the School of Pharmacy and CINJ as a research professor in medicinal chemistry, J Augeri has a storied history. He worked in the oncology space. He's done work with CINJ. He worked at BMS, and he essentially was the architect for Saxagliptin, which is a very exciting class of drugs to treat diabetes. By background, he has a doctorate in synthetic organic chemistry from UC Irvine, an NIH postdoc from Princeton, and he works very closely with us at the School of Pharmacy and at CINJ. And just a general nice guy all around, and a very smart guy. The other speaker, Brian Strom, also a smart guy and a nice guy, is our inaugural chancellor for RBHS, and his major research field is in the area of pharmacoepidemiology. Some folks call him the founder of pharmacoepidemiology, and he's done a lot of pioneering work looking at large databases sort of on the back end of drug discovery. So essentially, what's cool about this seminar today is that we have two individuals who essentially are looking at this compound from both sides of the drug development spectrum. And sometimes when we think of drug development, we think of the preclinical and then the human trials and then some post-marketing. But here we have the very early part and the later part once a drug is out. So that's how this whole idea started. David came to me and said, you know, the chancellor is doing a lot of research with this compound, working with the FDA and governmental agencies. I developed a drug. Don't you think it would be a great idea to put together a symposium where we will look at this very important class of drugs from both ends of the spectrum? So after several months of trying it, and now we are here, so it's my pleasure to first introduce J Augeri, who will lead us off with the Saxagliptin discovery story. David?
What I want to share with you today is the drug discovery story on Saxagliptin. A few important points about this program that made it possible to be able to identify a candidate that could go the distance and receive FDA approval. First of all, I think we had a really tight-knit group of scientists, both chemists and biologists, working together, communicating together to come up with not only design ideas for compounds, but also very good design ideas for assays that would yield really useful information to us chemists to allow us to prepare the next more advanced series of compounds. Second of all is it is an easy target to study in the sense that it's in the circulating blood volume. It's not inside the cell. It's more easily accessible for compounds that are prepared. And we used an approach where we developed a chemotype that had very good drug-like characteristics at the onset. Good bioavailability, good solubility, good chemical stability. And from a compound like that, we built in potency. I think you'll see that. With that, we'll get into some of the background biology. Primary defects in insulin secretion and building insulin resistance contribute to the etiology of type 2 diabetes. Diminished postprandial glucose or insulin secretion as a consequence of either defects in the beta cell or complete loss of beta cell function give rise to hypoglycemia and diminished sensitivity to insulin. At the time that we decided to look into this new mechanism, it was around 1998, 1999, the incidence of type 2 diabetes in the Western world had tripled since 1980. So just in that roughly 2-decade time period, this disease state became tremendously more prevalent. We decided to focus on the enteroinsular axis surrounding GLP-1, which is an incretin hormone. It's secreted from the L cells in the ileum, and it elicits an insulin response from the beta receptors because it's a very potent GLP-1 receptor agonist on the beta cells in the pancreas. It also triggers a response from the alpha cells on the pancreas that essentially decrease the glucagon peptide from sending a signal to degrade stored glucose in hepatic and muscle tissue, which is glycogen. Glucose is stored in a very polymeric form called glycogen. And so with those types of traits kept in check, one sees the natural feedback mechanism for maintaining glucose levels in normal, healthy individuals. Unfortunately, GLP-1 is very short-lived. It has a short half-life of 60 to 90 seconds circulating because dipeptidyl peptidase 4, located proximal to the L cells in the brush borders of the intestinal tract, can clip the 30 amino acid peptide, which is GLP-1 7 through 36, to generate an inactive product, which is actually a weak antagonist of the GLP-1 receptor, GLP-1 9 through 36. And so it was our thought that targeting inhibition of DPP-4 would extend the circulating time of GLP-1 and counter rising glucose levels in type 2 diabetics. DPP-4 is a 766 amino acid protein composed of 2 symmetrical subunits. It is membrane bound as CD26, located in the brush borders of the intestinal tract, and the kidney and the liver, in the pancreatic duct, and in vascular endothelial cells. A protease that sheds the 29 amino acid N terminus of this bound protein liberates the free form of DPP-4 that now is free to circulate in the blood, in the circulating volume. It clips the penultimate amino acid at the P1 prime amide bond to produce this dipeptide, shown here if it's alanine, and again generating the weak antagonist 9 through 36. P1 can be alanine or proline, but it has to be one of those 2 amino acids. The P2 function can be any amino acid or amino acid surrogate. In the late 90s, the landscape surrounding some of the early phase programs targeting DPP-4 show cores or molecules that you see here as reversible, the thiazolidine or some of these proline nitriles, as well as some irreversible inhibitors, the boronic acid from Point Therapeutics or this phosphonate. Our goal at BMS was to generate a reversible inhibitor that could be given once a day. And it needed to have enhanced solution stability. There are issues with the compounds that you see here that give rise to very short-lived lifetimes in circulation due to some stability issues, which I'll go over in more detail in just a moment. But this Novartis molecule here was published in 1999, 2000, with what is in effect a proof of principle study that shows that this molecule inhibited DPP4, and you can see a very statistical significant decrease in plasma glucose as a consequence of treatment with this agent. So this really prompted us to move quickly and step up the pace of our program, as we now knew there was some real competition out there. The solution stability is one issue we decided to focus on first, thinking that we could build potency into our program over time, but we certainly needed a compound that was going to be stable in solution. And the problem with the terminal amino group in this dipeptomimetic is that nucleophilic attack on the proline nitrile can lead to this cyclic amidine, which very quickly hydrolyzes to the diketopiperazine. And these diketopiperazines are side products that you might see in typical peptide coupling reactions. It's at the bottom of a thermodynamic well, and they're totally inactive. So we needed to have solution stability associated with our molecules for any chance of moving ahead with a viable compound. We could restrict the conformation of the molecule to avoid the conformational flexibility that would allow for this nucleophilic attack to generate amidine, but those cyclic systems really weren't very active at all. In fact, most of them were over 10 micromolar. However, the other type of conformational restriction would be to flatten out the five-membered ring and thereby extending the peptide chain in a more linear fashion, such that there'd be less of an incidence for this terminal amine to direct nucleophilic attack toward the proline nitrile. So we decided to use some precedents that Steve Hennessy had actually introduced the year before in looking at captopril, and he generated both cis and anti bicyclic systems with this methanoproline analog. And in fact, in this case, it souped up the potency of the compounds. That would be a good thing if that did that with our systems. We're mostly interested in having a flatter system. And in 2003, the Novo Nordisk group published a paper showing, in fact, that the normal envelope conformer that one would observe in a five-member ring had indeed been transformed to a more flatter conformation as a consequence of the bicyclic methanoproline bridge. And not only is the methanoproline bridge important for that conformation to be more linear and to prevent nucleophilic attack by the proline, but the alpha center, what is the chiral center on the P2 substituent, if it is a beta-quaternary center, then it has a much more sterically bulked up R group that also prevents the terminal amino group from attacking the nitrile. And you can see even in, well, with the essentially terbutyl R group on the P2 substituent with the 4,5 cis proline, you see a 45-hour half-life. It gave us great confidence that we had a chemotype and some information about the R group that in the P2 substituent that would be leading us toward optimization of these by peptide compounds. So, and even with the terabutyl group on a proline lacking the cyclopropyl group, you can see that that beta-quaternary center has an impact on the stability of the molecule at physiologic pH. We made both the 4,5 methanoprolines as well as the 3,4 methanoprolines, and we studied every isomer of those. But the cis arrangement turns out to be the most potent of the possible stereoisomers. With the 4,5 system, we made them where we dehydrated this prolamid with first reducing it with L-selectride and then performing the elimination, and then through a zinc carbanoid insertion achieved the methanoproline bridge. The diastereoselectivity of the reaction when the R group was ester was 4-to-1. We had hydrolyzed that in the research group to the acid to get 9-to-1 selectivity, and that's pretty much what we scaled up most of our core for our further SAR studies with. But actually, to go one step further and make the amide, as our process group showed us, we could achieve 100-to-1 selectivity. And so this is a fairly straightforward approach toward making the core of our system. Clipping the Bach group would set this up for peptide coupling with the P2 substituents to be able to generate the final products for enzyme analysis. Producing the 3,4 methanoproline was a bit more involved. We followed the work by Sasaki, where this sulfone was deprotonated with two equivalents of N-butyllithium. First deprotonation of the Bach, followed by deprotonation of the sulfone to generate an anion that would attack the triflate of this glycidyl triflate, a reactive little devil there indeed, to generate this epoxide intermediate. An ion transfer would yield then this hydroxymethyl cyclopropane, and then mesylating this alcohol to generate a leaving group, followed by deprotonation of that mesylate would allow the pyrroline to close, and then simple deprotection and reductive removal of the sulfone, followed by Jones oxidation, gave us our desired core. So a lot of synthesis goes into generating a series of compounds for SAR, but what we found was that the two compounds, again, that proved to be most potent, now we're moving away from solution stability and now measuring enzymatic behavior and generating KIs, that the 4,5 and the 3,4 methanoprolines were optimal. Again, we went with the 4,5 system because the solution stability was about double that of the 3,4. And we were confident that we could certainly enhance the KIs well past a 25 nanomolar compound. But at the outset, we had a compound that had pretty good drug-like characteristics in terms of having a clean P450 profile and negative activity in the Hurg assays, good membrane penetration, and good bioavailability in all the species that we looked at. So we began to take this system and combine it then with functional groups that in in essence, pointed toward a compound with a very branched beta-quaternary center. And while the terabutyl or teraleucine showed 7 nanomolar Ki inhibition, when we had a ring like a cyclopenyl or methylcyclopenyl R group, the more extended branching then began to tell us that the P2 pocket was rather large in size and very hydrophobic in nature, and a more branched beta-quaternary center with a larger ring system was perhaps the way to go. But we had pretty much tapped out of all of our commercially available systems. It was time to produce some non-natural amino acids. Borrowing on the methods of Kazmaier, then we produced ring systems like the ones you see in this system, in this blue box at the bottom, that are generated from a 3,3 Claisen ester rearrangement. The starting molecules toward accessing the precursor for that rearrangement involve taking the triethyl phosphonoacetate and deprotonation with sodium hydride to generate the ylide, and condensation with this ketone, where, remember, this could be any kind of ring size or substituted ring, would give these alpha, beta, unsaturated esters. And then reduction of those esters to the alcohol and condensation with glycine would set up the precursor for the 3,3 rearrangement, where deprotonation with LDA at this carbon, followed by a zinc-mediated Claisen rearrangement, gave these non-natural amino acids that had a lot of branching at the beta-quaternary center. These products were racemic in nature, but coupling to an antimerically pure core allowed for the formation of a mixture of diastereomers, which were easily separable. And just to spare you, the D amino acids were all above 10 micromolar, and the natural L configuration is what you see here, was most active. This molecule in particular is one that I'll point to that we found quite attractive. And at this point, I'm going to tell you a little bit about an assay that I thought was crucial in being able to identify a truly active compound. Anytime that you can tie PK to PD is going to be very helpful in establishing a compound with good bioavailability and good efficacy. Well-designed assays of that nature are extremely powerful in choosing active compounds for a program, and not always easy to pull off in every program you come across, but it's always worth trying to imagine how one could design such an assay. In this case, the assay involved rats, Zucker rats. We would use 6 in each dose group, and we would dose compounds at 30 minutes at 0 time frame and take blood samples at 30 minutes and 4 hours. The day before we performed these experiments, we would cannula the rats with a jugular cannula such that blood samples could be easily removed with minimal manipulation on the day of the experiment. The compounds were orally administered, and then 30 minutes and 4 hours following dosing, the blood samples were taken and they were analyzed to be able to determine the extent of DPP4 inhibition in vivo. But all the assays were performed in an ex vivo sense, so we called this the ex vivo rat model. but we were dealing with taking a look into the in vivo activity of these molecules. So that was a very powerful assay. We were also able to look at glucose and insulin levels through taking those blood samples and the traditional biomarkers that made that are certainly characterize the biology of diabetes. What we found when we administered this compound was that the pharmacodynamic half-life was lengthy. However, the parent compound was gone after by the 30-minute time mark. We no longer saw parent peak, although the pharmacodynamic effect extended beyond 4 hours and beyond. We saw a rapid turnover in human liver microsomes. It even showed some inhibition of CYP3A4. But looking at the olefin, and the first experiment that we did was one where I decided to just reduce the olefin and study that compound, and we didn't see the same behavior. So the olefin was highly suspect as a point where the molecule was being metabolized. Not only that, but unsaturated fatty acids in the body get broken up by adding water across the olefin function. So it seemed like a reasonable place to try to make analogs, attempting to make what we thought might be the putative metabolite. So it took a little bit of time to identify this molecule as a penultimate intermediate where I could perform oxidation reactions. I made the diol directly. That was very, that lacked potency, and it was, and it was clear that was not the metabolite that we were looking at. But clipping that diol to make this hydroxymethyl analog resulted in a compound where the pharmacodynamic effect was virtually identical to this olefin, except it hung around as a parent all the way through the 4-hour mark. The slow metabolic turnover rate was indicative of the fact that now this was no longer being metabolized. And again, we never proved it was the putative metabolic end product, but or metabolite, but the pharmacodynamic effect was nearly identical. And the CYP3A4 activity that we saw initially, we mixed this olefin with CYP3A4 and ketoconazole as a normal substrate. Those assays are usually run, and they read the assay at about 10 minutes. So we just decided, Ashish Khanna, one of the guys in the DMPK group, and I mixed these together, and we let the assay run for 30 minutes and 60 minutes, and we found that actually the CYP3A4 activity regenerated. So it wasn't an inhibitor of 3A4, it was a substrate that was competitive with the 3A4 substrate. So that was another clue that metabolism was going on with this compound. So we continued to make compounds that were more beta branched at this quaternary center, and we decided to move up and make one that was much more highly branched, and like this adamantyl system. And we saw the exact same kind of profile with this as we did with this cyclopentyl in the sense that it had a rapid metabolic turnover and had poor bioavailability, although it had an excellent inhibition of DPP4 at 30 minutes and 4 hours. In fact, it inhibitions in excess of 85 to 90% in vivo at the 4-hour mark, and it wasn't dropping at all from the 30-minute time point. So we thought we had a molecule that was way more potent than just 5 times the Ki would indicate. And my associate Dave Betaquaternary, who made this compound, and I'll show you how he made this P2 amino acid. It was through an asymmetric Strecker synthesis, and he actually took the amino acid he made and applied some oxidation conditions that existed in the literature toward oxidation at the bridgehead. I was hoping that it would oxidize at the bridgehead. I didn't know that before I asked them to go down to the library and check that out. This is the days when you went and you beat up the stacks looking for a paper. You didn't go online and take literature off of online journals. And he was extremely good at finding stuff that most people would never find. Not only that, but he was an excellent experimentalist as well. I was very interested in seeing this bridgehead hydroxyl get oxidized from a design standpoint because it has C3V symmetry. So as long as you see oxidation at that bridgehead, then you don't generate a new chiral center. And that was something that I valued a great deal because I didn't want to get beat up by the process guys when they found out if we advanced a drug with yet another chiral center that they had to synthesize at kiloton quantities. And the fact that it was a hydroxylated adamantane that had no chirality was a beautiful thing. Dave Betaquaternary was able to make this hydroxylation happen from the exact same literature conditions of the plain adamantane ring on the amino acid, which I had doubts would hold up to those harsh basic conditions, but in fact it did. This molecule is Saxagliptin in the end. And this is why we're so excited about being able to do this in such short order. It was about 2 weeks from the generation of just the plain adamantyl compound to getting the saxagliptin molecule. And the asymmetric Strecker reaction begins with taking an ester, first reducing it to the alcohol, and then oxidizing up to the aldehyde and condensing the 2-phenylglycanols, a chiral auxiliary. And then through the methods of Strecker, you add in a nitrile that you hydrolyze to generate an amino acid. And then you simply clip the amino acid or the chiral auxiliary from the amino acid to give you this non-natural adamantyl amino acid as a product and antimerically pure. These are the conditions that existed in the literature with just a plain adamantyl group, and they actually worked on the Bach amino acid, which we're very gratified by seeing. And in an hour, we could, in 51% yield, we had an amino acid that we could couple to our antimerically pure proline core. So we had identified a compound that had a very clean profile in every respect, and we thought that we had a candidate we could move ahead with. The kinetics of binding essentially show that there's a very slow on rate, as the adamantyl is probably very close to being the size of the P2 substituent or the P2 pocket, and so water needs to be displaced that is located in that pocket in order to afford binding there, and then it delivers the proline nitrile, which undergoes nucleophilic attack by DPP4 as a non-classical serine protease. You can see that the serine 630, and here's your catalytic triad, your SER, HIS, and ASP, is non-classical in the sense that usually the classical serine protease is SER, ASP, histidine, but these two are reversed. Nonetheless, they perform their general acid, general base type of duties and deprotonate the serine, which then adds nucleophilically to this nitrile of the pyrrolidine of our core to generate a covalent acyl enzyme complex. That's what gives this molecule the potency in vivo that we see, because it takes some time for the acyl enzyme complex to break up, releasing the drug and liberating free enzyme. This is the Exview assay that I was talking about, and you can see that in this case we're measuring DPP4 inhibition in rats, where 6 rats are dosed with 4 micromolar of compound, which is about, for the molecular weight of Saxagliptin, which is like 315, about 1.2 mg per kg, roughly. And that you can see that the inhibition seems to top out at 80%. With this assay, that's pretty much where we think we can max out. Use a fluorogenic histidine proline AFC. AFC is a 7 amino 3-trifluoromethylcoumarin as a fluorogenic substrate. And so the amino acid gets clipped, releasing the coumarin moiety, and that in a fluorescence, with reading fluorescence absorption and excitation, you can do linear regression methods to calculate your degree or your percentage of inhibition. And so we could also, looking at blood that was taken from these animals, look at, again, at glucose and insulin levels as well. And so that you can see that in these normal rats, when we look at GLP-1 levels, you can see that at 0.5 hours or 30 minutes, you see the potentiation now through DPP4 inhibition of that circulating GLP-1. This is available now to be able to elicit a signal from the beta cells in the pancreas to secrete more insulin. And because you now take DPP4 out of the picture, you have more sustained circulating levels of GLP-1 to generate the insulin levels you need and counter the postprandial hypoglycemia, or hyperglycemia rather. And you can see that at 4 hours, you still have essentially the same degree of DPP4 inhibition, and your levels of GLP-1 are still quite high. At 16 picomolar levels, that's about 3 times the Ki, or the Ka, I should say, of GLP-1 for the GLP-1 receptor, because GLP-1 is between 5 and 10 picomolar, probably more like 5 picomolar. And so this is roughly at 4 hours 3 times the Ka, which is really covering the required levels for sustained insulin secretion. And then in a glucose tolerance test with Zucker rats, you could see that at 4 hours, you could see the difference in controls versus treated are quite different. And while those of us who know a little bit about the numbers for milligrams per deciliter, and still over 200 looks a little high for most of us. The Zucker rats, I think they weigh about 3.5 or 4 pounds. They're the biggest, fattest rats you've ever seen. And when I first saw these rats, I was afraid of how much compound we were going to have to make for the studies. I was thinking rats, yeah, okay, 200 mgs, that'd be fine. And then I saw the rats, and I was thinking those things look like dogs. So anyway, we eventually made enough to cover the studies, and you can see that in this case, in a dose-dependent manner, 1 log unit difference. You could see that we continued to lower glucose levels as we increase in dose. Although in some cases, and you'll see later on in the human studies, that you sort of don't need to go to the top dose to achieve the highest level of efficacy. It sort of tops out and levels out, sort of in accord with what you see here regarding the plasma DPP-4 activity. You can see that as you advance in dose, once you get past 0.3 micromoles per kg, which is essentially about 0.1 mgs, 0.1 to 0.2 mgs per kg, you don't really see any further inhibition of DPP-4. So you know you're at your sustained levels of GLP-1 at that point. And the insulin sort of shows the same thing as we go from 1 micromolar to 3 to 10 micromolars per kg. You see that increase, but as the efficacy studies will show, and this was done in OB mice, in humans you won't have to go that high. So the overall profile of the compound is shown here, with a pretty striking Ki of 0.6 nanomolar, very good molecular weight at 315, with the hydroxyadamantyl, where I showed you 45 hours, a little less than 2 days for that T-butyl, 4,5-methanoproline. Now with this hydroxyadamantyl, you see a solution stability of double that, and so we had retained the drug-like features that we were pursuing. And importantly, we had good selectivity for the other DPP-4 enzymes, straight out 400-fold against DPP-8, 75-fold for DPP-9. This was a little bit more of a concern. We wanted more selectivity than that. DPP-8 and 9 are involved in T-cell activation and immune responses. We would rather see 1,000-fold selectivity over those if we could. But those are intracellular peptidases, and Saxagliptin doesn't really get into the cell very efficiently. So it stays in the circulating volume. Some of the other enzymes that have homology with DPP-4 that we don't want to hit, we aren't really accessing. So in a practical sense, in the end, we're achieving pretty decent selectivity. The ADME profile shows good selectivity or good activity in terms of its bioavailability, over 50% in 4 species. We don't inhibit any of the P450 enzymes. And in multiple species, we see the readouts that we seek to see that allowed us, with the safety that was performed, to have advanced this compound to clinical trials. So moving on to the clinical efficacy summary, just a few slides here. Here's the monotherapy at week 24 in humans, you can see that now we're looking at HbA1c levels, so it's sort of a more of a long-term log sort of approach of looking at serum glucose, where now this is glycosylated hemoglobin. So you get a much more time average look at serum glucose levels, because glycosylated hemoglobin levels are a direct reflection of more of an average of glucose levels in the blood over time. And with a 95% confidence interval versus p-values, confidence intervals are better to look at. They include all the data. P-values, you can sort of cut the data off where you want to get the best p-value you're looking for. But with a confidence interval, you're including all the data. And it sort of shows that really at 5 mgs as a dose, which is somewhere around 0.1 mgs per kg for humans, we're achieving about a 0.5 H1B reduction, and that's about what we expected to see, and what the other DPP-4 inhibitors like Saxagliptin or Januvia achieve. But we actually achieved quite a considerably higher decrease in HbA1c when we combine it with metformin, which really modulates glucose levels that derive from muscle and hepatic tissue. And so the combination treatment that's available with metformin is called Onglyza, and it is a more popular treatment, I think, because the combination of Saxagliptin and metformin bring you down to 2.5 HbA1c units below your starting blood levels, and that is a substantial drop in HbA1c. So the open questions, sort of, I alluded to a little bit, where we did see a dose-related decrease in absolute lymphocyte count, because we perhaps were interacting with some of those other enzymes that had homology with DPP-4 that could affect T-cell activation. Not sure, but ongoing clinical studies, which Dr. Strom will allude to, I'm sure, address this issue. Not clinically significant, as we saw at the outset at the end of phase 3, but that's why you have phase 4 studies, because they're a lot longer and they reveal a lot more. And of course, there is a label, I think, that all the DPP-4 inhibitors have regarding keeping watch on the cardiovascular health of some patients who have both diabetes and heart disease. So here are the 5 approved DPP-4 inhibitors on the market. The Merck compound, Vildagliptin, was discovered by Novartis. Interestingly, it has the hydroxyadamantyl function. There was some discussion about this in the literature following the publishing of our paper and first disclosure of our compound. I think you could see how we arrived at our compound in a more stepwise discovery way. And this compound was made through a library fashion where there was alpha-chloroamides that were substituted with a whole library of amines to generate these kinds of substitutions, and they happen to have the amino hydroxyadamantane in their library and wound up with this compound. This was never approved in the U.S., but it was approved in the European Union. And Takeda was produced Acknowledgements just a couple years ago, and 4 years ago, Boehringer Ingelheim introduced linagliptin. And so one that I'm leaving off because it wasn't approved yet is omaragliptin, which is a new compound that just got approved in Japan 8 days ago, which is a new chemotype by Merck, and it's a once-a-week therapy. So that'll be interesting to see how the FDA looks at that. I have my reservations about inhibiting that target over a week unless it has exquisite selectivity. I think the once-a-day daily dosing is an achievement, and you sort of let the activity of the DPP4 return so that it can act on its normal substrates. But if you take out for a week, I'm not sure in the long term how that's going to turn out. So it leads me to acknowledge a few people. Dave Biederbenner, my associate, who is a really bright guy, a tremendous experimentalist. Larry Hammond and Mark Kirby were the group leaders for the program in the late stages. They took saxagliptin, that molecule, and moved it on through advancing it to phase 1 and phase 2 trials. Jeff Roble and Rex Parker were the chemistry and biology group leaders that led the beginning of the program and all the way through about to the discovery of when we discovered saxagliptin. And of course, this whole team of people and a lot more people not mentioned is what is required in order to see a drug through its discovery and development all the way to FDA approval. So with that, I'd like to thank you for your attention, and I'll take any questions.